In this presentation, we are going to study biochemically important carbohydrates. Here are our agenda: monosaccharides, disaccharides and oligosaccharides, polysaccharides, and dietary considerations regarding carbohydrates. First would be some biochemically important monosaccharides. D-glucose, which is also called dextrose or blood sugar or grape sugar. Ripe fruits, particularly ripe grapes, are a good source of glucose, which is often referred as a grape sugar. Dextrose is an optically active D-glucose in aqueous solution. It rotates plain polarized light to the right. And it is considered to be a blood sugar from the fact that blood contains dissolved glucose. So it is also the most abundant in nature and most important on a human nutritional standpoint. It is actually used by our cells or the cells of living organisms as a primary source of energy. D-glucose as a blood sugar. Actual glucose concentration in the blood is actually dependent on the time that has elapsed since the last meal was eaten. So, for its normal concentration, it is about 70 to 100 milligram per deciliter of blood, and it rises to about 130 milligrams per deciliter after the first hour of eating. So the concentration will decrease over the next 2 to 3 hours, going back to the normal range. A 5% mass per volume glucose solution is often used in hospitals as an intravenous source of nourishment for patients who cannot take food by mouth. So for example, if you get admitted in the hospital and you would have an IV insertion, this is the liquid that goes straight to your veins to avoid being dehydrated and as your energy source, because the body can use it as an energy source even without digesting it. Next biochemically important monosaccharide is the D-galactose. So as you can see on the upper right part of the screen, that is the Haworth projection structure for D-galactose. It is synthesized from glucose in mammary glands for use in lactose in the human body, and it can be converted into glucose. It can be found in dairy products, sugar beets, and other gums and mucilages. So sometimes it is also called the brain sugar because it is a component of glycoproteins found in our brain and nerve tissues. It is also present in chemical markers that distinguish various types of blood. Next would be D-fructose. D-fructose is a basic component of table sugar, and biochemically it is the most important ketohexose. It is also known as levulose and fruit sugar, which is found naturally in fruits, some vegetables, and honey. It can also be found in corn syrup, and sometimes it can be used as a dietary sugar because less is needed for the same amount of sweetness. So if, for example, you buy beverages or other processed foods and you look at their ingredients and you find HFCS, so it pertains to high fructose corn syrup, which is used to sweeten these products. Another biochemically important monosaccharide is D-ribose. It is a component of a variety of complex molecules which include our RNAs and DNAs and also the energy-rich compound such as ATP or the adenosine triphosphate. Disaccharides and oligosaccharides. Before we discuss some important disaccharides and oligosaccharides, let us first discuss how disaccharides and oligosaccharides and polysaccharides are being formed. So let us start with the disaccharides. Now, in disaccharides, it is composed of two monosaccharide units connected by a glycosidic linkage. So these two monosaccharides, one will act or will function as a hemiacetal, so the one on the left, and the other one will function as an alcohol. When these two condenses or combine together, so the reaction is a condensation reaction, a glycosidic linkage is formed. So it is a bond between two monosaccharides resulting from the reaction between the hemiacetal carbon atom alcohol group of one monosaccharide and an alcohol group on the other monosaccharide, together with a condensation reaction leading to the formation of disaccharides. Water will be the byproduct. Now, first, for our biochemically important disaccharide, we have maltose. As you can see from the image depicted on the screen, maltose is a disaccharide composed of two D-glucose units. Maltobiose or malt sugar is another term for maltose, so it is produced when starch breakdowns. The breaking down of starch takes place when seeds germinate and in us humans during starch digestion. It is also a common ingredient found in baby foods and in malted milk. Here shows how maltose is formed. So you have an alpha D glucose and another D glucose whose configuration can either be an alpha or beta configuration. When these two glucose units condenses, an alpha 1 to 4 glycosidic linkage is formed. So if you notice, glycosidic linkages have specific names. So this one has an alpha 1,4 linkage as its name because the configuration of the monosaccharide on the left is an alpha configuration, and then what is being connected by the glycosidic linkage is the carbon number 1 from the hemiacetal monosaccharide and carbon number 4 of the other monosaccharide. The glucose unit on the right of maltose can open and close, so it is in equilibrium with its open chain aldehyde form. So what does it mean? It means that there are actually three forms of the maltose molecule. So you have the alpha maltose, the beta maltose, and the open chain maltose. In the solid state, the beta form is the one that is dominant. The most important chemical reaction of maltose or disaccharides in general is hydrolysis. So what is hydrolysis? That is simply the reverse of condensation or the reverse on how disaccharides are formed. So in hydrolysis, the glycosidic linkage is hydrolyzed with the presence of water and an enzyme, and it will be hydrolyzed into its component monosaccharides, which this time is 2D glucose units. Here are some food sources of maltose. So we have sweet potato, honey, peaches, spelt, which is a species of wheat, bagels or a bread roll, which originate in the Jewish communities in Poland. Traditionally, it is made from yeasted wheat dough, and we also have bread. Then broccoli has maltose as well, molasses, which is a viscous byproduct obtained from the refining of sugarcane or sugar beet juice into sugar. And we have food products sold in the market, like for example, Cocoa Crunch and Milo and other energy bars which contain maltose. Cellubiose, another important disaccharide, is produced as an intermediate when cellulose undergoes hydrolysis. So if you notice, it also contains two D-glucose units, and the other one it must have a beta configuration. Now, the change in configuration on the D-glucose on the left results into a beta 1 to 4 glycosidic linkage. It is a reducing sugar, and it also has 3 isomeric forms in aqueous solution. When hydrolyzed, it will also produce 2 D-glucose molecules. So if you notice structurally, cellubiose and maltose has a lot of similarities, but because of the difference in the configuration of one of the glucose molecule components, it will also result in a different glycosidic linkage, which will result to these two disaccharides behaving differently from each other. Now, the enzyme that breaks the glycosidic linkage in maltose, which is maltase, is an enzyme that can be found in both humans and in yeast. However, the enzyme that can hydrolyze the glycosidic linkage of cellubiose, which is cellubiase, is an enzyme that cannot be found neither in humans nor in yeast. So which means that we cannot digest cellubiose and it cannot be fermented by yeast. Lactose is made up of a beta D galactose unit and an alpha D glucose unit, which is joined by a beta 1 to 4 glycosidic linkage. Now, this is a reducing sugar, and this is a major sugar found in milk. In mammalian mammary glands, the enzymes take glucose from the bloodstream and then synthesize lactose in a four-step process. Lactose is an important ingredient in commercially produced infant formulas that are designed to simulate mother's milk. Now, the souring of milk is caused by the conversion of lactose into lactic acid by bacteria in milk. So, pasteurization is done, which is a quick heating process that would kill most of the bacteria and will retard the souring process. Lactose can be hydrolyzed by acid or by the enzyme lactase, forming an equimolar mixture of galactose and glucose, meaning when lactose is broken down by the enzyme lactase, it produces an equal amount of galactose and glucose. In the human body, the galactose produced is then converted to glucose by other enzymes. So we know that it is the principal carbohydrate in milk. And as comparison, human mother's milk contains 78% lactose as compared to 4 to 5% lactose found in cow's milk. Here are foods rich in lactose, so cow's milk and goat's milk. Then we have milk byproducts like yogurt, kefir, cheese, and ice cream. Lactose intolerance and lactose persistence. So what do we mean by lactose intolerance and lactose persistence? For many people, the digestion and absorption of lactose are a problem. So take note that normal lactose intolerance is actually not considered to be a food allergy situation, because when we say food allergies, it actually arises when one's immune system responds to a foreign substance or tagged by the immune system as an invading allergen. Lactose intolerance does not actually involve the immune system. So it is a condition wherein people lacking the enzyme lactase, which is needed to hydrolyze lactose to galactose and glucose. Here are the estimated prevalence of lactase persistence in various populations. So as we can see from this diagram, we have Sweden as having 99% lactase persistence, Scotland, so Denmark. These are countries whose products are milk-based and they consume a lot of milk products. Another important disaccharide would be sucrose, or what we know as table sugar. It is from juice of sugar cane and sugar beets. And sugar cane is 20% by mass sucrose. So sucrose is the most abundant of all disaccharides and occurs throughout the plant kingdom, while sugar beets contain 17% by mass sucrose. So sucrose is composed of two monosaccharides. So we have an alpha D-glucose and a beta fructose connected together by an alpha beta 1 to 2 linkage. Now sucrose, unlike maltose and cellubiose and lactose, is what we have seen from the previous slides, their structure. Sucrose is actually a non-reducing sugar. Sucrase is the enzyme needed to break the alpha beta 1 to 2 linkage in sucrose, and it is present in the human body. So that is a reason why sucrose is an easily digested substance. When sucrose is hydrolyzed, it produces again an equal mixture, an equal concentration for the mixture of glucose and fructose, which will now be called an invert sugar. So when sucrose is cooked with acid-containing foods like, for example, fruits or berries, what takes place is partial hydrolysis, forming some invert sugar. So jams and jellies, which are prepared in this manner, are actually sweeter than the pure sucrose added to the original mixture, because one-to-one mixtures of glucose and fructose taste sweeter than sucrose. Here are some sugar substitutes that is currently being marketed or circulated. So we have saccharin, sucralose, aspartame, and neotame. Among the sugar substitutes, saccharin is oldest of the artificial sweeteners. It has been in use for more than 100 years. It is 300 times sweeter than sucrose, and examples of how it's marketed. It is named as Sweet and Low and Sugar Twin, both saccharin-based commercial products. Questions about its safety arose in 1977 after a study suggested that large doses of it could cause bladder tumors in rats. So, as a result, the FDA proposed banning saccharin, but public support for its use caused Congress to impose a moratorium on the ban. So, in 1991, on the basis of many further studies, the FDA withdrew its proposal to ban saccharin. Another sugar substitute is aspartame, marketed as NutraSweet. It accounts for three-fourths of current sugar substitute use. It is 180 times sweeter than sucrose. So it is used mostly in the United States and Canada. It has quickly found its way into almost every diet food on the market today. Aspartame, just like saccharin, are not heat stable, and therefore it cannot be used in products that require cooking. The safety of aspartame lies with its hydrolysis products. So, when aspartame is hydrolyzed, it produces the amino acids aspartic acid and phenylalanine. These amino acids are identical to those obtained from digestion of proteins. The only danger aspartame poses is that it contains phenylalanine, an amino acid that can lead to mental retardation among young children suffering from phenylketonuria. Sucralose is a derivative of sucrose. It is heat stable, so that is its advantage with aspartame. It can be used in cooking food or in cooked food. It is 600 times sweeter than sucrose and is calorie free. Aspartame loses its sweetness when heated, while sucralose does not. It is calorie free and cannot be hydrolyzed as it passes through the digestive tract. Neotame, a general purpose sweetener, is already also heat stable. It can be used in a wide variety of products, including baked goods, frostings, frozen desserts, puddings, and fruit juices. Its sweetness is 7,000 times greater than that of sucrose, so only a small amount is needed to sweeten products. So its caloric impact is also negligible. It is aspartame derivative. Oligosaccharides. Oligosaccharides contain 3 to 10 monosaccharide units bonded to each other through a glycosidic linkage. Raffinose, the one on the left, and stachyose, the one on the right, are two naturally occurring oligosaccharides commonly found in cabbages, onions, broccoli, Brussels sprouts, whole wheat grain, and all types of beans. The monosaccharide components of the trisaccharide raffinose are galactose, D-glucose, and beta D fructose. Stachyose is a tetrasaccharide which contains an additional galactose unit as compared to the structure of raffinose. Now take a look at the glycosidic linkages present on both. Now, humans lack the needed enzyme to metabolize either raffinose or stachyose. When these oligosaccharides are ingested in food, they pass undigested into the large intestine where bacteria act upon them. This bacterial action is what causes discomfort and flatulence. In biochemical systems, it is more common to encounter oligosaccharides as components of more complex molecules rather than like raffinose and stachyose, which are in their free state. Human blood is classified into four types. So we have type A, type B, type 8B, and type O. Now, what determines the type of blood a person has is the oligosaccharide that is attached to the person's red blood cells. Now, the biochemical basis for the various types of blood involves oligosaccharide molecules that are attached to the plasma membrane of red blood cells. Now, these oligosaccharide attachments, which we would be calling biochemical markers, are of three different formulations. One is a tetrasaccharide, and the other two are pentasaccharides. Four monosaccharides contribute to the makeup of the oligosaccharide marking system. So one, we have alpha D-galactose. Then we have alpha L-fucose, alpha N-acetyl D-glucosamine, and alpha N-acetyl D-galactosamine. So of these four, one is a simple monosaccharide, that is the alpha D-galactose, and the other three are monosaccharide derivatives. Now note that all the oligosaccharide markers have a common 4 monosaccharide sequence in their structure. Now, what will determine the blood type is the absence or presence of a fifth monosaccharide unit. Type O, if we will look at the representation on the screen, lacks a fifth monosaccharide unit. Type A blood has N-acetylgalactosamine as a fifth unit. Type B blood has a galactose, fifth unit. And type AB will contain both type A and type B markers. If a blood transfusion is necessary and the patient's own blood is not available, then we look for donors, right? So the donor's blood must be matched to that of the patient. Blood of one type cannot be given to a recipient with blood of another type unless these two types are compatible. What will happen if there would be wrong blood type and the transfusion is done? So it can cause the blood cells to form clumps, which will be a potentially fatal reaction. Now in this table, we can see the compatibility relationships of the different blood types. So people, for example, with type O blood are universal donors, and those with type AB blood are universal recipients. Another oligosaccharide would be solanine. So if you will notice on the screen, these are examples, or these are potatoes. Now many plants, including potato, produce toxins as their defense mechanism against insects and predators. The small amount of solanine present in properly stored potatoes is not dangerous and actually contributes to the flavor of the potatoes. However, when there is an excessive amount of solanine, potatoes will taste bitter. Solanine amounts in potatoes increase when potatoes sprout and when they are exposed to sunlight. Improperly stored potatoes would have that greenish color which develops in their skin and the flesh under the skin. Such greening is a warning already that solanine levels are higher than normal and may even mean that it's approaching to an unhealthy level. The green color is not actually solanine, but it is chlorophyll. Sunlight exposure stimulates both solanine and chlorophyll production. So peeling potatoes deeply to remove the green color also removes excess solanine. Sprouts need to be removed and potato eyes need to be cut out. Sprouts also have high solanine content. So if you see your potatoes having sprouts already, so that means the solanine level is already increasing. So you must remove that so that it won't taste bitter and that it won't have any after effect. Deep frying reduces solanine levels while boiling is ineffective in decreasing such levels. The trisaccharide present in solanine is composed of the monosaccharides D-glucose, D-galactose, and a rarely encountered monosaccharide, which is L-rhamnose. L-rhamnose is unusual since it is the L isomer, wherein D isomers are commonly present in nature. And if you will look at the structure, it contains a methyl group on carbon number 6 rather than the CH2OH group. Now we move forward to polysaccharides. Unlike monosaccharides and most disaccharides, polysaccharides are not sweet and they will not test positive in Tollens and Benedict solutions for reducing sugars. They also have limited water solubility because of their very large size. Polysaccharides can also be called as glycans. What are the important parameters that distinguish polysaccharides? First, the identity of the monosaccharide repeating units in the polymer chain. So we have here the unbranched homopolysaccharides and the branched homopolysaccharides, unbranched heteropolysaccharides, and branched heteropolysaccharides. Now, what is a homopolysaccharide? So when we say homopolysaccharide, this is a polysaccharide wherein only one type of monosaccharide monomer is present. So for example, in an unbranched homopolysaccharide, all the monosaccharide components are that of glucose. Now, when there is branching, then we call that a branched homopolysaccharide. So, therefore, a heteropolysaccharide is a polysaccharide in which more than one, so usually 2 type of monosaccharide monomer is present in the chain. Next, the length of the polymer chain. So it varies from less than 100 to over 50,000 monomer units. And then the type of glycosidic linkage present between monomer units. The degree of branching of the polymer is also another important parameter that will distinguish polysaccharides from each other. The ability to form branched chain structures distinguishes polysaccharides from the other two major types of biochemical polymers, the proteins and the nucleic acids, which only occur as linear or unbranched polymers. What are the different types of polysaccharides? So we have three. We're going to discuss three. So we have the storage polysaccharides, the structural polysaccharides, and the acidic polysaccharides. A storage polysaccharide is a polysaccharide that is a storage form of monosaccharides and is used as an energy source in cells. So we have starch and glycogen. Homopolysaccharide containing only glucose monosaccharide units, that is starch. So starch can be branched and unbranched. Energy storage polysaccharide in plants. Now, if excess glucose enters a plant cell, it will be converted to starch and then it will be stored for later use. When the cell cannot get enough glucose from outside the cell, it hydrolyzes starch to release the glucose. Two different polysaccharides can be isolated from most starches. So that would be the amylose, which is the unbranched homopolysaccharide, and amylopectin, which is the branched homopolysaccharide. Amylose, unbranched, and usually accounts for 15 to 20% of the starch, and the type of glycosidic linkage present is the alpha-1,4. The number of glucose units present in an amylose chain depends on the source of starch. 300 to 500 monomer units are usually present in the amylose chain. Amylopectin, on the other hand, is a branched glucose polymer. It accounts for the remaining 80 to 85% of the starch. A branch occurs about once every 25 to 30 glucose units, and the branch points involve alpha 1 to 6 linkages. Because of the branching, amylopectin has a larger average molecular mass compared with the linear amylose. Up to 100,000 glucose units may be present in the amylopectin polymer chain. The starches present in potatoes and cereal grains account for approximately 2/3 of the world's food consumption. One test to identify the presence of starch is the iodine test. Iodine is often used to test for the presence of starch in solution. So starch-containing solutions will turn dark blue-black when iodine is added. Just like what you have observed when you did the iodine test or the starch test in the biochemistry laboratory. Another storage polysaccharide which is found in animals is glycogen. So its structure is somewhat similar to that of amylopectin. Glycosidic linkages present are alpha-1 to 4 and alpha-1 to 6. It's 3 times more highly branched compared to amylopectin, and it is much larger with up to 1,000,000 glucose units present. So its function is similar to that of starch in plants, and it is sometimes referred to as animal starch. When excess glucose is present in the blood, normally, for example, if we eat too much starch, the liver and muscle tissues will convert excess glucose to glycogen, which will then be stored in the tissues. Now, whenever the glucose blood level drops, like for example, when you are doing exercise or when you fast or even by doing normal activities, some stored glycogen is hydrolyzed back into glucose. Structural polysaccharides. These are polysaccharides that serve as structural elements in plant cell walls and animal exoskeletons. So we have cellulose. This is the structural component of plant cell walls. Unbranched glucose polymer. Glucose residues in the beta configuration. So the glycosidic linkage present is a beta-1 to 4. The woody portion of plants like the stems, the stalks, and the trunk, they have particularly high concentrations of this fibrous water-insoluble substance. So a cellulose polymer chain contains about 5,000 glucose units. Cotton if we would look at its component, is almost pure cellulose, so around about 90 to 95% cellulose, while wood is about 50% or 35 to 45% cellulose. Cellulose fibrils are formed due to extensive hydrogen bonding within and between cellulose chains. So these are insoluble in water and are quite strong and rigid. Even though it is a glucose polymer, it cannot be a source of nutrition for human beings. So the reason for that is because we lack the enzymes capable of catalyzing the hydrolysis of its glycosidic linkage. Now, even grazing animals, they also lack the enzymes necessary for its breakdown. However, found in their intestinal tracts are, like for example, the horses, the cows, and the sheep, they contain bacteria that produce cellulase, the enzyme that can hydrolyze cellulose glycosidic linkages. Now, the intestinal tracts of termites contain the same microorganisms, which enables termites to use wood as their source of food. So despite its non-digestibility, cellulose is still an important component of a balanced diet because it serves as a dietary fiber. Another structural polysaccharide, so these are for animals or the insects, we have chitin. Structurally, chitin is identical to cellulose, except that if you would look at it, the monosaccharide present in chitin is a glucose derivative, so which we call the N-acetyl D-glucosamine or NAG, rather than the D-glucose itself. So it's probably the second most abundant organic compound on Earth. It is a structural homoglycan found in the exoskeletons of insects and crustaceans, and also in the cell walls of most fungi and many algae. The product of complete hydrolysis of chitin is D-glucosamine. It is marketed now as a dietary supplement which is claimed to help with joint problems. However, there are also claims that D-glucosamine decreases joint inflammation and pain associated with osteoarthritis have yet to be substantiated. Acidic polysaccharides are polysaccharides with a disaccharide repeating unit wherein one of its disaccharide components, so this is a heteropolysaccharide, is an amino sugar, and one or both disaccharide components has a negative charge because of the presence of a sulfate group or a carboxyl group. Two of the most well-known acidic polysaccharides are what we are going to discuss, which are hyaluronic acid and heparin. Hyaluronic acid, so if you will look at the structure, it is a heteropolysaccharide because there are two monomer components. So the structure contains alternating residues of the N-acetyl beta-D-glucosamine and D-glucuronate. Highly viscous hyaluronic acid solutions serves as lubricants in the fluid of joints in our body, and they're also associated with the jelly-like consistency of the vitreous humor of the eye. So hyaluronic acid have a glass-like appearance. D-glucuronate is a carboxylate ion formed when D-glucuronic acid loses its hydrogen atom. So hyaluronic acid can be found in our eyes, in our skin, and in our joints. So today, hyaluronic acid is very famous for being a component of cosmetic products. Last, we have heparin. Heparin is a small, highly sulfated polysaccharide with only 15 to 90 disaccharide residues per chain. So its repeating unit contains D-glucuronate 2-sulfate, and N-sulfo-D-glucosamine 6-sulfate. Heparin is known to be a blood anticoagulant, so it's naturally present in mast cells and released at the site of injury. So mast cells are part of the body's immune system. They play an important role in the healing of wounds and the defense of our body against pathogens. They are more abundant in tissues that commonly come into contact with the outside world. It prevents the formation of clots in the blood and retards the growth of existing clots within the blood. However, it does not break down clots that have already been formed.